This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded on February 1st, 2016. I'm Kevin Bonham, and with me this evening are Kate Franz.
Hello.
Sounding lovely with her new non-broken microphone.
Oh my goodness, it's great. I'm going to be so wonderful. I'm going to sound so clear, and it's going to be all because of this microphone, and also because I'm not drunk.
And your teeth are all in?
Yeah, still currently I have all my teeth.
Yeah, so,
But does one of them have a giant hole in it?
Currently, yes, one of them does have a giant hole. It's very sad.
Did you end up crying? You promised not to cry.
I didn't cry, and let's see here. I'm trying to think. So, like, I've just had increasingly traumatic experiences at the dentist this past week.
Uh-huh.
So
Sorry to hear that.
I had to go back. I got yelled at by a dental hygienist who asked me if I grew up in a developing country because my teeth were so bad. She assumed I had never had dental care and that I did not grow up around fluoridated water.
Wow. All right.
Yeah. So then I yelled at her, and then she gave me the most painful tooth cleaning ever.
I feel like there's a lesson in there somewhere.
Yeah, somewhere. Maybe one day I'll learn it.
That is terrible.
That's really awful.
But it probably wasn't as bad as the root canal, right?
No, I don't think so. My root canal was actually almost a pleasant experience. My endodontist was really funny, and I talked to the hygienist who assisted about her son going to grad school. So it was as comfortable as it was going to be, I think.
Okay, excellent. Well, despite the fact that it's February 1st and we're recording, and Kate promised to be drinking on this episode,
I know, I'm sorry.
Turns out that she stayed at work, and so she's continuing her dry January into the 1st of February.
Actually, that's not true. I went out. So yesterday was my and Jordan's 4-year anniversary. So we went out to go get dinner, and I had a margarita with dinner, and it was lovely.
Oh.
But I think that is okay.
Yeah. February is a short month anyway, so I'm pretty sure that has something to do with it.
Yeah, just give February the last day of January. Yeah. I think that's perfectly reasonable.
Yeah, that's fair.
Matt, I saw what you were drinking, but you should share it with us.
Yeah, I've got actually a Yingling today. Yingling regular. So this is actually a wedding beer. We had this beer at our wedding, and Emily at the time said we should get Yingling Light because we're going to have a bunch of leftover beer, and I don't like regular Yingling. I like Yingling Light. And I said something that ended up not being true, and so now we have a whole lot of Yingling that I drink sometimes. It's the moral of the story.
I distinctly remember loading giant cases of unused alcohol into your father-in-law's car after your wedding.
Yeah, those cases are now on the refrigerator that I'm looking at right now, longingly.
Yeah, so they made the trip from Ohio, I guess, huh?
They did, yeah, yep. So we drank a whole lot of whiskey the other night. Probably way too much whiskey the other night. So anyway,
good. I wouldn't want any of our listeners to think that you weren't an alcoholic. That's good.
Right, exactly. Yeah. Well, I was sober last time, so here we are.
Were you really? I thought you were.
No, I think no, not the time before. It was vodka, and then
that's the most recent episode that I edited. That's why I'm
right. Yeah. So
I'm one episode behind.
We're on again, off again, but today's beer.
Okay, phenomenal.
Yep.
I love Yingling.
Yingling's good stuff.
Yingling's great. If you're not from the East Coast, they actually just started selling Yingling in Boston, maybe like a year or two ago. But so if you're not from the East Coast, you may have never heard of Yingling, and it's not spelled the way that you think it is.
Nope.
But it's pretty great. It's like as cheap as Coors and Budweiser, but actually tastes okay.
I was gonna say it's like if you take Coors Heavy and you make it just a touch sweeter. That's what Yingling tastes like.
Interesting.
That sounds gross, and I would not drink that. So I'm gonna have to disagree.
So it really does taste like normal Coors. I've had like a blind taste test because In lab, John's favorite beer is Yingling, followed by a close second of Coors, and they taste identical. They really do. So if you like Yingling,
That
you
makes
like
me
Coors.
unhappy.
Sorry.
All right.
I love Coors. I will die for light brewed beers all day long. I
do drink
love
a fair
them.
amount of Coors Light. Yeah.
I love Coors Light. I love Bud Light. I love Bud Light Lime. I love
Oh God.
I love Bud
no,
Heavy.
you take that back.
I love Coors Heavy. I love PBR. I love all those beers. They have drinkability, baby. Drinkability. And I am for that all day, all night. IPAs,
And
disgusting. Tastes like you're drinking
Audiommunity.
perfume.
Brought to you by beer. Beer. It's what you
Terrible beer, apparently.
Wonderful,
I may have
delicious,
to just-
drinkable beer.
I think I may just beep out Kate's entire last rant, just make it seem like she was cursing the whole time, because that's what it felt like to me. Just have the noise sound.
Right. I
for
mean,
that whole
alternatively,
rant.
we could just start contacting major brewing corporations to see if they'll give us money.
Or beer, honestly.
Right.
We'll settle for beer.
That's true.
This episode brought to you by, well, for me, this evening, Hogarden. We're doing free advertising. We probably shouldn't, but oh well.
I'm fine with
this
it.
This is a Hogarden Vit that I found in the immunology fridge downstairs. All of the good beer is gone, so I think before the next recording, I'm going to have to go.
You still have magical appearing beer, so I don't want to hear your nonsense.
That's true. But I'm going to have to go buy a case of something good.
Yeah.
The problem is I can't store it in the fridge downstairs because it will disappear. That has happened to me more than once.
This is
I
what
feel
the
like I'm
mini
taking
fridge
this
was built
beer,
for.
but I paid for it.
Right. But I feel like...
Right, but
Yeah.
I can't get a mini fridge paid for in my office, and I honestly can't afford it because I'm poor.
Go to Target. There's like a tiny little one for like $40. Like because like all of the college students, they get all of their crappy little mini fridges. Or you
Right.
can Craigslist
Or
that
just-
in like 3 months when everyone moves and they have all these mini fridges that they can't take home.
Or just
That's
drive
probably true.
up and down Mission Hill on September 2nd.
That's disgusting.
Yep.
Anyway, okay, we're gonna continue on this evening.
Yep.
For the science portion of Audiommunity, we are going to be discussing a fun paper that is kind of an amalgam of a couple of other episodes that we've done, I feel. So the title is "The Intestinal Microbiota Contributes to the Ability of Helminths to Modulate Allergic Inflammation." And I'm pretty sure that we've touched on each piece of this title in previous episodes. So we've talked about the intestinal microbiota just a couple weeks ago. We've talked about worms and how they're weird in terms of the immunity that they induce. Not weird in the sense that it's anything more crazy than anything else, just that very few people actually study what worms do because they're kind of hard to study in mouse models. And nobody knows what the hell Th2 immunity is about.
Nope.
And then we've also discussed allergic inflammation. So we had the episode about, I think, bee allergies or pollen allergies. I can't remember now.
It was bee pollen.
Right, but the bee pollen was the weird hippie nutritional supplement thing. It was
Sure.
flower pollen and bee stings.
Bee stings, you're right.
I think it was bee stings.
Yep, it was
In any
bee
case.
stings.
So in this paper, basically they are combining all of those things and looking at the interaction with each other. And we actually didn't discuss before we started recording who was going to go through the introduction, but
I mean,
I'm just
go for it. Go for it.
I'm just going to roll with it, and then we can get into the figure. So the interesting piece of this that I think we've mentioned before is this idea of the hygiene hypothesis, which is this weird observation that people in countries that have much lower hygiene than the first world, so places that are exposed to a lot of infections, particularly parasitic worm infections, which thankfully in Europe and America, we don't really have to deal with that much. Those populations tend to have much, much lower rates of autoimmunity and inflammation. Or I'm sorry, autoimmunity and allergies.
Right.
And the correlation with worm infections is very strong. In fact, there are people that are looking into ways of treating autoimmune disorders and allergies with parasitic worms, either worms themselves or products of the helminths.
And we've talked before about this a little bit. We talked about the MS studies, for example, with people plus with MS, essentially plus or minus helminths, and you can actually see a decrease in emerging MS symptoms in patients that have been exposed to worms. So there's a lot of cool biology going on here that, like you said, we've touched on before.
Right. And there are people developing. I mean, it makes sense to me that you might treat inflammation in the gut, like IBD, with worm infections, because most of these worm infections are pathogens of the intestine. But it still seems crazy to me that somehow worms being in your gut can alter autoimmune disorders or allergies in other locations, like in the brain for MS, or in the case of this paper, we're going to talk about airway inflammation. So these are going to be worms in the gut of the animal that is somehow influencing the immune responses in the lung, which is not really directly connected except by like blood circulation. In terms of location of the immune system, it seems kind of weird that you would have these this distant action.
But we also see other examples of the helminth immune suppression and immune responses in other parts of the body. I was just thinking about when I briefly talked about the worm infection that correlated with increased fertility. And so the hypothesis there was that the worms suppress the immune system, which makes like the uterus more responsive to accepting like an implantation of an egg, which was pretty interesting.
Yeah, and in the other direction, I think, and this was in the pregnancy and immunology episode that we published a couple weeks back, probably a month or two back now. But you can also look, and this immune suppression is not necessarily a positive thing in all scenarios. So there is a negative correlation with things like malaria, TB, pertussis, Hep C, HIV even. So, there is this sort of general immune suppression. And for things like allergy and pregnancy, that might be a good thing. But for other infections that require a strong inflammatory response, having this worm infection and broad suppression might be a negative thing.
Right. So the other component of this story is the intestinal microbiota. So these are the bacteria that typically hang out in the gut of all mammals.
Well, not just bacteria. Bacteria, archaea, viruses, fungi, other single-celled eukaryotes. There's lots of crap in there.
That's true. The bulk of it, at least in mammals, is bacteria, though.
The bulk that has been identified is bacteria.
Yeah, I'm going to have to agree with Kate here. There's, it's probably the largest biomass, but there's a lot of other stuff going on.
Sure. Okay. So just excluding viruses for just a moment, because detecting viruses is a bit harder to do, or not harder, but it's not usually detected with the methods that are used to investigate the intestinal microbiota. But we can get a pretty good picture of all of the cellular life that hangs out in the gut, and a very, very small fraction of it is not bacteria. There is archaea, there is fungi, but there's even some eukaryotes. What's up?
There's even some eukaryotic life that seems to be stable across. So and I mean, this, looking at microarrays all over the place, I agree with you completely that the greatest diversity is certainly going to be in bacterial components in the intestine, but that doesn't mean that a couple non-bacterial populations have a profound effect on the way that we view this problem.
That is entirely possible. They do exist. People haven't spent a ton of time studying them because they're such a small fraction, I think, of the overall pool. I think people haven't focused on them as much.
But what I've been meaning to tell you guys is I'm a virus.
That doesn't make any sense.
Okay.
Although, did you guys see the paper recently that showed that all the people that were constantly claiming the 10-to-1
yes, I love
bacteria
that.
to human paper, they're basically like, that's bullshit.
Yeah, that was great.
Turns out it's more like 1-to-1, and it also changes every time you take a dump.
I was going to add that point. That was my favorite part. Was like, if you have a hearty enough poop, you might swing it back till you're mostly human. I was like, oh, that's lovely. Thank you.
That is very nice. Also, I'm sure most people that listen to this podcast have also been exposed to NPR, but on the way home, NPR did a little blurb on cesarean section babies. C-section babies, and
that.
being able to sort of reconstitute with mom's microbiome through the application of vaginal secretions, actually, immediately after birth. Mm-hmm. So anyway, maybe we'll talk about that some other time.
Yeah, this has been one of my favorite sort of like conversation killers at dinner parties, especially when there are pregnant women around, is like, if you get a C-section, you really should dip your child in feces.
Right.
Just to make sure that you reconstitute their microbiota, and so to prevent allergies and inflammation, because it turns out that babies born via C-section are much more likely to get allergies and inflammation. Or sorry, I keep saying that. Allergies and autoimmunity than babies that are born through the normal, through the vaginal canal.
Like and me. I was a C-section baby, but I have no allergies. You know what's funny? My brother, so I was a C-section, but my brother was not, and he had such severe asthma as a child. I know that this is anecdotal, but I always found that so sad. Like, you would thought, like, I was like the smaller baby. I like didn't have shoulders. I was a C-section baby. I was really early. No, like not sickly lungs at all. My brother was like a month late, super huge, like really big baby. Natural birth, like, oh God, my poor mother. But he was like so sickly. Anyway, the end.
Sorry, Kate's brother.
Yeah.
He's good now.
You got screwed on the genetic lottery there, apparently.
He has better teeth than me, though. He has rocks in his head.
Right. In any case, so it's clear that the microbiology, or I guess the biology generally, of the gut actually has fairly systemic effects on the immune system. And I suppose that this makes a kind of sense. I mean, we know that, for example, your gut is a generally immunosuppressive environment because you don't want to make immune responses against every foreign protein that you ingest. Also, inflammation in your gut tends to cause problems because it turns out that eating food and digesting it is pretty important. So if you have massive inflammation throughout your gut, it's going to cause problems. So there's probably been a strong selective pressure to prevent rampant inflammation in the gut. But I think it's a little surprising.
Just as an aside, that doesn't mean that you should eat an alkaline diet, people. That is all complete BS. Sorry, just had to like insert that.
People do that? What is that?
People believe that like
this is great. You haven't heard about this?
The addition of inflammation, inflammation, the idea of it in the gut is tied to eating acid or like. Oh. So okay, yeah. So people think that like if you eat like acidic foods, that is a pro-inflammation state for your gut. So in order to fight inflammation, you have to eat neutralizing foods or like things like alkaline food. So like water with like a slightly lower pH, really like dark leafy vegetables.
Higher pH.
Oh, higher.
Okay.
I think we actually went through this discussion on here before about my ability to distinguish pHs. But I meant lower concentration of hydrogen ions.
Of course you did.
No, I absolutely did. You can ask anyone in lab.
pH has pH is a log scale, so
it's a negative log
values
scale.
Are right.
So what's the pH of stomach?
2.
Yeah. Okay. And those things are still connected, right? The stomach and the intestine.
Yes.
Not very directly,
but there is a neutralizing
just with a hole between one and the other.
But there is like a neutralizing, like all the stomach acid is neutralized right as it enters your intestines. So you don't have to worry about that.
So we just mentioned that that's on a log scale, right? That pH value. So how much pH 8 stuff would you have to eat in order to neutralize your stomach effectively?
Matt, we're talking about woo here. So why are you trying to introduce logic into the conversation?
I would just like anybody that's thinking about this to think about the math for just a second. And then if you still don't believe it, do a quick experiment. Just eat Tums all day, every day, and then call us and see what happens and report back.
Well, you'll probably stop pooping.
Well, right. On the upside, you'll become less and less human.
True. Very true. Well done. Nice callback there. Okay, so we know that the stuff that goes on in your gut. Okay, so there's a few things that go into this paper, I feel like, in terms of introduction. So first is we know that worms will decrease symptoms and prevalence of allergies and autoimmune disorders.
Correct.
We also know that the intestinal microbiota has an effect on systemic immune responses. And one of the things that was discovered a couple years ago is that, so we've known for a while that the balance of intestinal microbiota in very broad terms, if you just look at 16S sequencing of the bugs in the gut and you say like there are more firmicutes versus Bacteroidetes bacteria in the gut, this can have an effect on the overall inflammatory state of the body. So it turns out that people that have metabolic disorders, things like type 2 diabetes, they'll have a prevalence of one of these broad classes of bugs versus the other. I never remember which is the pro-inflammatory and which is the anti-inflammatory. The same correlations happen between the C-section births and the natural births. So we know that the shape of the microbiota will affect systemic inflammation. And one of the ways that was discovered a couple years ago that this happens is through the production of these short-chain fatty acids, SCFA. And so this is things like propanol and butyrol, small organic compounds that are secondary metabolites of some bacteria and not others. These short-chain fatty acids can have an anti-inflammatory effect, not only in the gut,
this
but
is why you gotta eat your fiber.
Systemically.
This is why you got to eat your fiber, because bacteria digest the fiber and then they make short-chain fatty acids. And
yeah, and the fiber selects for the bugs that are able to do that metabolism.
I feel like fiber should be redefined if that's the case, because I feel like fiber has always been known as the stuff that you don't use and can't digest. And
well, you don't digest it.
Okay, fine, Kate.
There is caloric content, but I think the idea is that, most fiber does not get converted into calories, but some of it is converted into these short-chain fatty acids, and these short-chain fatty acids can have a marked influence on the immunology of your entire body. So that's all known going into this paper. And so these authors... I mean,
to sum that up, would be infection with worms is like immunosuppressive. And having a functioning like a full microbiota, so like you don't clear it out with antibiotics, that also leads to more or to less allergy and less autoimmunity if you have an actual microbiota. So like worms, less allergy, microbiota, less allergy. Are they related? Is it like the same mechanism? Is it a related mechanism? Do they actually interact? I would guess that's what I would say, like the hypothesis going into this paper is.
Yeah, exactly. We have these two things, both of which hang out in the gut, and both of which can have dramatic impacts on inflammation, in particular allergies in this paper.
Yeah.
And so, the question is, are they related? Do they interact with each other? And it turns out the answer is yes. So that's what this paper is about.
Yes. Through an interesting mechanism, which I feel like is not fully defined in this paper.
That is definitely true. Though
we found this knockout mouse, it worked.
Yeah, though it is. Though I mean, like, not to overly criticize the paper. It is a really interesting paper, but
it is a good paper.
Everything
past short chain fatty acid, I was just like, okay, sure.
Yeah. Although I have to say, even that much is like. People have known the hygiene hypothesis has been around for like 40 years.
Yeah.
And people have been like getting people to ingest eggs from whipworm to treat their IBD, which is like the crudest way of making this work in a clinical setting. So if we can figure out anything about how this is happening, maybe we can figure out ways of treating allergies and inflammation, or God, I keep doing that, allergies and autoimmunity better. And so I think this paper moves that along in ways that I think hasn't been moved along in quite some time.
So
can we find a paper to post with this, hopefully, where they just fed people a whole bunch of short chain fatty acids to see if it has a similar sort of effect?
Or, I don't know people, but definitely mice. There was a paper published from a lab here at Harvard a couple years ago.
Did that work? I assume that it did work.
Yeah, it's Wendy Garrett's work, I think.
Right. So So
Short chain fatty acids, good for the colon. That's what that's how they should be sold at Whole Foods.
yeah, I'm sure that they already are sold there under that exact catchphrase. But
if not,
it would be nice. It would be nice if there was like
that.
a
It's mine. You can't
setting.
use it.
That's not how trademarks work.
I believe you just have to declare trademark.
I declare
Okay.
bankruptcy.
I don't know that reference.
That's from The Office.
The Office is
Okay.
great.
All right.
Okay, so
We're having more digressions than I feel we normally do.
on this paper. Okay, so a very, very short summary would be that worms work in collaboration with bacteria to create short chain fatty acids, which seem to have some sort of effect on the immune system. Okay, with that hypothesis, the idea that you could just feed people, so people who have autoimmunity or some sort of allergy likely would have some sort of messed up microbiota. And I wonder if the addition of only worms really would help. Because if you don't have the particular bacteria there, or you don't have a strong enough selection for them, how helpful is the worm really going to be if you don't, if you only have one half of the collaboration? How helpful is this treatment even going to be?
Well, I mean, this paper would suggest, because they sort of test that, not exactly, but they test that basically by giving these mice tons and tons of antibiotics, right?
Yes.
And so you start with a mouse that will get the dust mite sensitization, right? And so under normal conditions, you have, you basically give huge doses of dust mite antigen. That's essentially how this sensitization works. But a normal mouse will become sensitized and develop this sort of allergy. And then if you give that mouse helminths, then all of a sudden the mouse doesn't get the same sort of allergy that it did before. But if you wipe out the microbiome through these broad spectrum antibiotics, that helminth effect kind of goes away. So I think the answer in a complete ablation system is sort of addressed there. But your question of, an altered biology rather than an absent biology, I think would be a little harder to address.
Well, we know that people that have allergies and autoimmune disorders have different microbiomes than healthy people in general.
Sure.
Yeah.
The problem is we also know that the bacteria themselves can be causative. So we know that if you, for example, take a generally inflammatory microbiome and you put it into a mouse that has a healthy microbiome, you can exacerbate problems. But the etiology is not super clear as far as I understand. So in other words, we don't know if people with allergies and autoimmune disorders have altered guts because of their inflammatory disorder, or if they have an inflammatory disorder because they had an altered gut microbiome. And the causation is probably very reciprocal. There's probably sort of interactions both directions. But maybe we should actually walk through the paper and bring this up when it comes up, because I feel like Matt mentioned dust mites and a bunch of stuff.
Sure.
So we should actually probably go through it.
That's the first figure. I was going to say first image. I don't know why.
So in the first figure, they have a model of airway inflammation that, as Matt mentioned, is basically you take house dust mite allergen or antigen and you sensitize the mice to it.
Right.
I think you just inject huge amounts of it, and it it's
do.
inflammatory by itself, and it induces inflammation.
And it's just smashed up dust mite bits.
Oh,
Right.
cute.
There's not a specific they're not making a recombinant protein. It's not a clean system. It's just a mashed up bunch of dust mites, and then you inhale a lot of it, and if you keep inhaling a lot of it, you get sensitized to dust mites.
Right. And so in this first figure, they do a thing that I really hate in papers like this, where they give the treatment first and then they induce the disease, which is kind of irritating. Mm-hmm. But in any case, they infect these mice with a helminth that's a mouse parasite. I need to go look at. So it's I'm gonna mangle this. It's Heligmosomoides polygyrus bakeri, or HPB for short. All of that is just hard to say. It's the worm. I'm just going to call it the worm because I don't want to say that. I think
the worm is good.
Okay. So they infect it with this worm, and then they sensitize to dust mites, or they just sensitize to the dust mite allergen. And what they find is that if these mice have been previously infected with the worm, they have less eosinophil infiltration into the lung, and they have less inflammatory cytokine production in the lung. And less
you might
pathology.
you might be asking, what are those eosinophils doing? And once again, none of us know the answer.
Yeah. Although you can go look at the awesome comic that Matt drew for the bee pollen episode. And it's funny. So infected with worm, you get less inflammation. This figure is sort of recapitulating information that's already known. I think they're just establishing this particular model system. I don't know if anyone had combined this worm with allergic lung sensitization before, but they're basically showing this system works. Yeah. So the worm infection reduces inflammation in the lung.
Yeah. And can I just throw out a technical thing here, just in case anybody is an immunologist or biological scientist and is everyone ever looking at antibody titers, please don't ever report antibody titers as OD values from an ELISA. So an ELISA is a way where you can basically say how much antibody against a specific thing an organism has. And the way that the assay works, the most direct readout is this thing called an optical density, where basically a machine is just reading literally how yellow something is. But how yellow something is doesn't actually relate to how much antibody is in any given system. You need like a normal standard curve, a positive and negative control, all of those things. The only thing that you can ever say looking at two OD values is that the two groups are different in some way, but you have no idea based on that whether they are 10,000 folds different or 2 folds different, depending on where they are on that standard curve. So the reason I bring it up is because they use OD values all the way through this paper, and there's a couple spots where it's actually really important to know just how different those titers are, and I have no idea.
Yeah, so, I 100% agree. That bothered me too.
Yeah. Anyway,
it's like I've seen someone
because I wasn't paying attention.
I've seen people report like qPCR values as CT values, and that similarly irritates me because you just have to put it in the exponent, bro. Come on.
I just believed that they were picking somewhere on the linear portion of the curve.
I mean, it could very well be
maybe
that it's a 2 folds difference, but the truth is that, antibody responses are usually over 6 logs. So if it's a 2 folds difference, then it's actually not that biologically important. Anyway, I'm sure
the question I had about biological relevance was this IL-4, IL-5, because that seems to be the most convincing data. Well, the IL-5 over the IL-4, just because you see in the Helminth treated mice that they have control levels in response to this antigen. Is this a good readout?
Well, the
does this mean anything interesting?
Typically, Helminth infections are going to be inducing type 2, so Th2 immunity, which is the type of immunity that you get IL-4 and IL-5 produced. So it is weird to me that in the worm-infected animals, they actually have reduced IL-4 and IL-5.
Interesting.
Because you would expect that the worm infection would increase those cytokines. Now, they're looking in the lung, not in the gut where the worm is.
Exactly. The problem with that is the eosinophils also release IL-4, IL-5 in response to allergy inflammation. So you really, to answer your question, I don't think that is great. It's not a wonderful readout because both things should be doing that. But I think a better readout probably is the pathology. And I imagine what happened here was they ran some flow, they saw that there were eosinophils, and then they did the pathology. And then someone asked them, are those eosinophils doing anything? Gotcha. And so they did these IL-4, 5 ELISAs.
And actually,
but they're not great.
Even a better readout is figure 1F, which is kind of a weird thing. So this is you treat the mice with this thing called methylcholine, and this is a standard assay that's used to look at airway inflammation. And basically what they're looking at is the difficulty that the mice have in breathing after treatment with this methacholine, which is kind of an irritant. And basically, low resistance means
they're breathing
no
fine.
inflammation, right?
Yeah.
That they're breathing fine. High resistance is that they're that it's hard. The inflammation is essentially restricting the airways and making it more difficult to breathe. And in this case, it looks like the worm infections are having no effect on the ability of these mice to breathe. So they've still got essentially the allergic inflammation, even in the presence of this worm, which kind of goes against I actually hadn't really noticed that. I was sort of comparing two different things there. Or maybe, no, that's weird. I'm confused now.
Yeah.
Because this is basically like invalidating. It feels like it invalidates the whole paper.
No, is the control not a negative control? Like we didn't treat with
the control is
this drug.
So the dust mite is like the HDM. So there's an HDM plus worm, there's an HDM no worm, and then the control is, I believe the control here is we didn't treat them with anything.
Right. So the control is that
that should just be
just get the
normal
methacholine.
breathing. Yeah.
And so they get higher doses of methacholine.
Oh,
it's an irritant, so it makes it hard for them to breathe. But in the presence, when they're sensitized to the house dust mite, they have a much harder time breathing.
Yeah. Yeah, I don't understand what they're trying to show there.
Yeah. It should be different if they're reducing allergy symptoms.
I think that's a reviewer experiment.
And like I said, it feels like it invalidates the whole paper. So now I'm very confused.
I don't think that it invalidates the whole paper. I mean, so we've talked about this before. So I'm not entirely sure what sort of like sensitivity is that dust mite supposed to be inducing, and which sensitivity is the methacholine. So are they like testing two different sensitivities?
It could very well be. Maybe it's just that the methacholine is different, and so
no. This is a standard assay of allergic inflammation. Is methacholine resistance. So what you're saying is even in the presence of helminths, you're still getting allergic sensitization. Yes.
Has anyone gone back and actually read what they say about that?
Yeah,
Nope.
It just says they just report it.
Infection did not impact airway hyperreactivity as measured by increased airway resistance after methacholine challenge. Period.
But they say, however, there were fewer inflammatory cells in the lungs after the worm infection.
But who gives a shit if there's less inflammatory cells if they still have allergic inflammation?
No,
right,
I mean
guys,
they
paper
they
over.
do show,
We're done.
They do show pathology, and the pathology. Yeah, I'm always.
But who cares about the pathology if the symptom? I mean, like what we care about in people that have allergies.
I never care about pathology. I'm sorry, Matt. I only want the things that I can measure with a computer.
I mean, I would argue that you can measure those things with a computer, but.
You can, but again, like what matters
subjectively.
Like, for me, functional readout is like all the cell type stuff, and it doesn't invalidate the whole paper because it is interesting. The worms are clearly affecting short chain fatty acids and all of these other readouts. But in terms of actually understanding effects on allergies,
right? But one could imagine that what we're testing here is an acute scenario, right, where we're seeing. The short-term increase in inflammatory cells, we're seeing a short-term increase in IgG1, we're seeing a short-term increase in pathology, and if we carried this out for a year, we'd have a long-term accumulation where there would be a significant change in the lung function.
I also just wonder if this is the wrong sort of stimulus. Like it's a more subtle thing. Maybe this is a better model if we were to look at like IBD or something.
Yeah, maybe.
Not that mice really get IBD, but like, whatever.
No, we can give them IBD.
Sure.
You feed them a detergent. They get real bad IBD.
Yeah.
Turns out if you feed mice SDS in their drinking water, it totally rips up their gut.
Because I think like the better data comes with looking at, okay, what is different about the bacteria when the worms are here?
Sure.
Yes.
and like looking at how, I mean, I think the strength of the paper is really in like, as Skip Virgin would say, like the trans kingdom interactions. So interactions between species within different kingdoms of life, so like worms versus bacteria, and like how are these two things interacting and changing the phenotype of like the host or the phenotype of the actual communities.
Right, and that nicely leads into figure 2, where they try to address that by basically knocking out, and this, Kate, is what you were talking about before, you take mice and you knock out their microbiomes by these broad spectrum antibiotic courses, and then you try to see if the infection with the helminth without the bacteria now in the gut still has the same effect on house dust mite. So essentially they use exactly the same methods, including the same reported OD values in IgG1 response. And they look at, whether the antibiotic ablation has an effect. And sure enough, it does. Although, I would say that the antibody data in this figure, unfortunately, becomes totally undigestible due to a lack of standardization there. But I think probably for the eosinophil data, there's still certainly more eosinophils when there are no bacteria present in the gut than when there are bacteria present in the gut. So clearly the helminths are having some effect through the bacteria on eosinophil infiltration.
To reiterate, what they're doing, they're sensitizing the mice to dust mite, and they see eosinophils infiltrating the lung. And then they treat with the worm, as in figure 1, and they get a reduction in eosinophils in the lung. And then they do the same thing, but they treat the mice with antibiotics before giving them the worm infection. And you get as much, or in fact more, eosinophil infiltration in the lung, whether or not you have a worm present with the sensitization. And they also do this other thing, which is interesting, where they co-house the mouse. And I think we've talked about this before, but mice are coprophagic, which is a scientific way of saying they eat each other's poop. And
poop eaters.
This makes it really, really easy to get mice to share a microbiota. So with humans, you got to do these complex fecal transplants. With mice, you just got to put them in the same cage, and their microbiomes will essentially equilibrate. And what they show is that if you co-house mice that have not been infected with worms with mice that have been infected with the worms, just the microbiota, which seems like it might include the worms as well, I don't know, but they're implying that you're just getting the bacteria transferred, and that is sufficient to reduce eosinophil infiltration, which is
I did wonder that.
Some worms have a different life cycle. It's not like directly fecal oral. So maybe that's the case here. I'm not sure.
Sure.
I didn't look it up, but I'm sure it's known.
Yeah. Somebody knows it. We don't. So that's figure 2. The bacteria are doing something.
Oh, wait. Of note, such co-housing allowed the transfer only of the bacterial microbiota and not of the parasite, because adult worms cannot replicate within their hosts, nor can they infect new hosts.
Oh, okay. Cool.
That sentence makes it sounds like worms can't grow or live or propagate themselves in any way.
Well, I think they all have such a lot of worms can have complex life cycles, so there might just be a stop. Like, it might not be the true host. So an infection can be established, but like...
Yeah, no, they give adult worms to the initial mice, and the adult worms are not the worms that will reproduce.
There you go. Worms are kind of gross.
Interesting. Problem solved, and also evidence that I do not read papers super thoroughly. So... moving on to figure 3. So here they're doing some stuff that we talked about in the last microbiome episode, which is they basically do these giant sequencing projects where they're looking at large clades of bacteria using DNA sequencing of 16S ribosomal RNA. And then they do a bunch of really fancy statistical modeling to show effects in ways that, even though I have been trying to read up on this literature for the last couple of years, still feels a little black boxy. Like, I know how to put in the commands in Chime in order to get these figures, but I still don't fully understand the algorithms behind them and what they're doing. But
that
ultimately,
doesn't seem problematic at all.
No,
I feel like figure A, it's just take the number counted and divide by the sum of everything counted.
True. That's
ridiculous, Kate.
That is true. Although actually it's
pretty good.
It's
algorithm.
A little bit more fancy than that, because there are biases in terms of what gets sequenced, and you have to normalize the reads, and there's a bunch of like fancy techniques to make sure that you're not duplicating stuff, and the algorithm development of these things is like an entire career in itself. But
how would we know if someone employed those fancy techniques or just added up and then divided by the sum?
Hopefully they would tell you in their methods.
Okay.
And hopefully a reviewer would be smart enough to ask for the calculation if it seems suspicious. I am not sure that happens all the time, though, so you should definitely be skeptical.
Well, this is why papers need to be reproduced.
That is true. But in any case, what we see is that when you infect mice with worms, their gut bacteria change. And this is actually not terribly surprising. What was surprising to me is how crazy high the Bacteroidetes bugs are in all of the mice. I feel like over 80% is not typical. And they have over 80% in all situations, although they only have I'm very confused by this figure. The way that they break things up seems very strange. But in any case, you see that the effect changes when you have infection with worms, which isn't terribly surprising. B is more interesting, and here they're looking at the proportion of these different short-chain fatty acids. So again, we knew previous to this study that short-chain fatty acid production is affected by the microbiome composition, and that short-chain fatty acids can have effects on the systemic immune system. And what they find is that you have increases in short-chain fatty acids, in particular acetate, which is a 2-carbon short-chain fatty acid, and propionate, which is 3-carbon short-chain fatty acid. Doesn't look like there's a huge change in butyrate, which is the 4-carbon chain, but
that's the one that everyone loves to look at too.
Right.
Yeah.
Butyrate is like the interesting one.
It's just clearly too long.
The thing that I thought was interesting though was like, it's not that big of a difference. It's like it goes from like 6 to 10 millimoles per kilogram. Is that a lot?
Yeah. I don't know. I didn't go back and look at the original paper from Wendy Garrett to see what effect sizes they were measuring. But that actually doesn't seem too off the mark. I would have to go double check.
I mean, I think it depends on where you're looking, and like local concentrations might be different. Yeah. Like super local concentrations might have an effect. So like broadly.
Right, yeah, because we're basically looking in the feces in this. So throughout the gut, you're going to have different concentrations of these things.
Yeah.
And maybe the reason we don't see effects on butyrate is because the butyrate is further metabolized. I'm not sure.
That is probably very likely, because butyrate is metabolized in the colon, and it's very important for cells to get their butyrate, and it makes them anti-cancery. That's what I know about cancer. Anti-cancery and pro-cancery.
Yep. I think that's what the alkaline diet's for. It's to get the butyrate all the way to the colon.
Yeah. Mm-hmm. Oh, yeah. Eat your oatmeal.
I think Kevin actually was just in pain when I said that.
Yeah, that hurt my brain. Even though I know you're joking, it still hurts. It's like if you punch someone in the face, but you don't mean it, it still is painful. Right. Okay, so they also do these principal component analysis plots, which I think I've mentioned before. I can explain what they mean, but they also seem like magic to me.
The two groups are different. Yeah. Naive groups versus worm-infected groups. They seem different in that their ovals are largely non-overlapping. That's how I know they're different.
Yeah, and again, like this does not seem surprising to me, that you would have... Worms are giant parasites that are squiggling through your gut. They're going to be consuming nutrients. They're going to be inducing inflammation. They're going to be secreting all kinds of things. Like, of course, that's going to affect your microbiota. So I feel like this doesn't really... I mean, I guess maybe the point is that it affects the microbiota in a consistent way, because the worm-infected bugs or the worm-infected samples cluster together apart from the naive. So I
Except
guess that
for the naives that cluster with the worm-infected ones.
yeah.
There's only like 2 of those, right?
There's 3.
Yeah.
Okay, fine.
I don't know. These analyses, unless there's super clear differences, these analyses always bug me a little bit.
I don't know. I disagree with you guys. I think that I think the clustering to my eye looks like there's probably a real difference there. Whether that difference is expected or not is a totally different argument, but
I'm not saying that I don't believe the clustering. I'm just saying that like PCA makes me sad because like you don't know what is in that axis of the graph.
sure.
You don't know
but
what is
but there is one, and we know that.
Yeah. I don't know.
Did I ever tell you my preferred statistics method?
Tell
It's
us.
to show 100 scientists the same piece of data with 2 groups and ask each one, do you think this is significant? And then when you report the significance, it's just the significance of that number of scientists saying, yes, I do versus no, I don't. And that's how I think that these should be done.
Yeah. The problem is you can't just say scientists because like the vast majority of scientists, I think, maybe I'm wrong. Maybe I'm just exerting some wishful thinking here, but I kind of feel like most scientists, if they were honest with themselves, don't understand the vast majority of statistics they look at. Like they might be able to tell you sort of what a p-value means, but I don't think they really understand what a p-value is.
No, I'd be looking for gut feelings. Like, actually, it's apt because the Iowa
like
caucuses are tonight.
Okay.
And in the same way that the Iowa caucuses this year are looking for gut feelings, I feel like I can pull gut feelings of statistics out of scientists.
The way that I like to do science is to have phenotypes that you believe without any sort of statistics. Like my phenotypes have logs difference in them. And I like that.
Well,
I don't.
La-di-da.
It sounds like you might have been trained in the
viruses.
lab of John Kagan because I feel like he pounded that into my brain as well. Like,
I've always been this way. I can go into like a deeper explanation of where this comes from, the scarring that occurred to me. But anything that's too close and needs statistics, anything that like if you can run all of the different MATLAB analyses on it, regardless of if they're appropriate to run or not, and like you get significant versus not significant, and you're like, well, I like this Tukey method better. I'm going to go with that one. That one made my data significant. If you can do that, don't work on it.
Stop doing what you're doing. Yeah.
Right.
Science is hard.
Science is hard enough without having to rely on statistics. And I actually feel like I understand statistics fairly well. I studied a lot of math in undergrad. Like I actually do understand like the underpinnings of a principal components analysis, and I don't want to rely on that shit for anything.
Well, you also don't have data sets that are large enough where you could do principal component analysis, though.
I actually hate large data sets. Because I feel like large data sets are hypothesis generators, and you still have to do basic science with them if you want to do anything interesting or say anything with any sort of like oomph behind it. So I hate large data sets. Oh boy,
Kevin, how do you feel about that?
I sort of agree with that. I mean, more or less.
I also really hate screens, but I'm going to do one, and I'm very excited about it.
Cool. I think that a lot of people draw too strong of conclusions from even like very like small data set experiments. Like you do a gene expression analysis of a cell type treated or untreated. A lot of people will extrapolate that out to like how those cells are going to behave in vivo, how it's going to behave in a particular disease. And I think that there's overinterpretation happening at all levels. Maybe it's more obvious in the large data set things, but anyway.
I like large data sets of single cell measurements, though.
Yeah.
I think those are a special kind of awesome, but...
I agree with that. Nicolas Chevrier is doing that now in his own lab. He's basically doing like treatments with LPS and various other stimulators, and then doing single cell and like RNA-seq analysis of like every single immune organ in the body. Yeah. Which is awesome and also a crap ton of work, and I'm really glad I don't work in his lab.
Well, Aviv's lab like published all the tech papers on that. I'm sure he was collaborating with her.
Yeah. Anyway.
Yeah. So stuff happened, and your bacterial populations don't look the same after you get worms.
Yes.
Yeah,
and
they're
also,
slightly different.
You can take the microbial populations that are induced by the worms, and then you can transfer them to other mice. So that's figure 4. And this is sort of recapitulating what we talked about in figure 2, but they just do it in a lot more detail, and they do all those fancy statistical methods. And here they do see an effect of butyrate and not of acetate and propionate, which is odd that it sort of flips in terms of which short chain fatty acids are produced, but whatever, we get increased short chain fatty acid production when you transfer the microbiota. And I think this is good news there are a bunch of companies now that are working on therapies, like drugs that are essentially just bacteria pills. So kind of like probiotics, but a little bit more controlled. And there's a company in Cambridge that's doing this now to treat C. diff infection. But I'm sure that they are thinking about other ways of taking beneficial microbiota and giving them to people in order to alter health outcomes. So
have you read these papers though that say how difficult it is to actually modify your microbiota composition? That's something I always think about with those sorts of...
So it seems to be short term, right? Like when you do, you can modify it, but it'll go back.
Yeah, exactly.
Right. Well, a big part of why it will go back is because diet has a huge impact. So the reason that C. diff treatments are so effective with fecal transplant is because that's an aberrant population of bugs that is usually induced by like long-term antibiotic treatment. Yeah. So if you give a normal microbiota and withdraw the antibiotics at the same time, you are basically just giving the opportunity to have a normal microbiota. Yeah. But I think that the idea that this company is going with, and I don't know a huge amount of details, but I think that they are looking at basically giving people giving them a nudge in the correct direction. And of course, if you stick with the same diet, if you have the same genetic background, which you are going to select for a certain... You probably will. You will select for a similar population of bugs. But I mean, if I can pop a poop pill that will make me thin, like maybe I'll just stay on the poop pill instead of having to alter my diet of candy and cheeseburgers.
I like my diet of candy and cheeseburgers. Thank you very much.
Yeah. So I'll take a poop pill if it keeps me thin.
I would be surprised though. I mean, you're referencing the study where they had my favorite animal experiment ever, because I'm not that hot on animal experiments either.
You are a very odd breed of scientist, Kate.
No, I'm a very common breed of scientist. I'm a biochemist. Biochemist. I like things in vitro. I like things that are like present and measurable and that I can like smush together and then separate. Like I'm a very old school sort of biochemist. But one of my favorite like animal studies was where they took disconcordant obese twins. So one twin was fat and the other twin was thin, and then they fed their poop to mice to see if the obesity was linked to the microbiota and if you could transfer obesity. And they saw that you could transfer obesity through the microbiota. And that in the mice, the microbiota that was dominant when you co-house the obese mouse from the obese microbiota and the thin mouse from the thin microbiota was that thinness was dominant, which was really cool. And so then they trace down to which bacteria is associated with the thinness. And they say it's like some Bacteroides species. But when you look at previous studies in humans, it's really dependent on race and gender and where you are where you live, like geographically, if that bacteria actually associates with anything positive or negative. So it's so confusing and so complicated. I would be surprised if it could be, I mean, like with most things, if it could really be boiled down to a pill where you could pop a pill and be thin. Like ideally, maybe.
On an unrelated note, I am starting a weight loss company focused exclusively on finding people's long-lost twins.
I would love to start a weight loss company where I bottle J-Lo's poop. Not bottle, but like I isolate the bacteria that are in J-Lo's microbiota, and then I will sell that. And then you will have your J-Glow, and you will also be like thin and a vampire because she is 45 and she looks 20. And I have also trademarked
plastic
this
surgery? Don't we have solutions
J-Lo?
for that?
Oh my goodness. No, if you said Kardashian, sure. J-Lo is a magical angel, and she is just like beyond human.
You should get in touch. You should get in touch with this company in Cambridge whose name I'm forgetting now. That is doing poop pills, and you should come to them with this idea that they should
that we sell celebrities, we
J-Los.
brand them by celebrities.
Yeah.
I have had this idea before. I'm actually not going. I've had like a separate idea. I'm not going to actually say it on this like recorded podcast because I have actually thought about starting a company with John Chow. But the celebrity
Another grad
branding,
student in the
I'm
Kagan lab.
very interested in. And then you just like you follow celebrity around, right? And then like you wait for them to go to the bathroom, and then you just hit that Starbucks restroom
That's
right
not
after
creepy
them.
at all.
No, I'm joking.
I
Everything
wonder
would
what
be
the
done with
ethics-
everyone's
we
consent.
had that ethics episode before. I wonder what the ethics of that would be.
The ethics
Like,
would be unethical. You have been judged and
Yeah,
ruled
I feel like-
unethical. The
I
end.
don't really.
Yes.
If someone goes and poops, and you collect their poop to harvest the
I mean,
microbiome,
Okay.
it's weird. It's
You-
like the
it's
senator
weird.
that.
No, don't get me wrong. It is
Right.
totally friggin' weird and crazy.
It's like the senator that took the Pope's water after he finished talking and then like brought it back to his office. And he's like, I've been saving this to give special people to drink. He just like snagged the guy's water and now is just hanging on to it in his office.
But that doesn't
It's
seem
weird.
unethical to me. It's
It-
weird.
no, I
Totally.
agree.
No, don't get me wrong. And I honestly don't think you're going to sell JLo's poop. I think more people would find. Well, I guess there are probably
I
a
think
small
she would.
subset of people that would pay a crap ton of money for it. No pun intended. I honestly didn't intend that.
I don't believe
So maybe you
you.
can make a business from it. Well, we've moved fairly far afield from our discussion.
Yeah, let's go back to short chain fatty acids.
Yeah,
Fatty
but I feel like
acids with less than 6 carbons.
there's not a ton more. Honestly, the rest
that
of
was
this
the good
stuff.
part.
Yeah,
We hit
the
the
rest of the
high.
stuff like
Yep.
FR3, I don't really care. Honestly, they find some gene that turns out ameliorates this effect, like that they're saying is responsible
Sure.
for the effect.
With no real explanation as
Yeah.
to
No
how or why
This is-
that gene might be important.
Yeah.
This is the problem. They're looking at a molecular question, and so they're like, I think this receptor is necessary. And then
Yeah.
they take a full genetic knockout in vivo model. Who knows what that receptor does? Probably a million different things depending
Right.
on the tissue, the organ. So it's really hard to say that there's a molecular connection when you're looking at the organization and the functions of tissues and organs during this sort of
Sure. Now, it could very well be that this is
this
true.
is why I don't like animal studies.
Right. It could very well be, though, that FR3 is truly a critical link. But I think that's probably best left to the next paper that they publish.
Yeah, so let's
I
just
agree.
say
I
basically
agree.
what they do is they repeat the stuff that they did in figure 2 where they used antibiotics to wipe out the microbiome. And here what they're doing is in the mice, they're knocking out a particular gene, this receptor FR3, which I don't even remember what it does, to be perfectly honest. It's some
It's
GPCR
a G protein, coupled receptor that responds to something. I think it actually responds. I think it actually does have something to do with short chain fatty acids.
Okay.
We'd assume.
Well, clearly, again, maybe I'm not the only one that doesn't read these papers super thoroughly, but
I do.
basically they knock out FR3 and
It is a
basically they
receptor
lose
for
the
short
effect.
chain fatty acids.
Thank you. So that I guess makes sense that if short chain fatty acids are actually causing this
be-all,
effect,
end-all.
then this receptor should be important for that, and it turns out that it is.
Right.
That's figure 5.
I guess I'm more interested in how the helminth is causing a situation where the bacteria are producing more short chain fatty acids. Like that is the mechanism that I would really like to
Yeah,
see.
and this gets back to
the-
Kate's question from the very
basic-
beginning, right?
I think the basic mechanism that they imply is that when you have the helminth present, you get this change in the microbial community. So you get like a different proportion of bacteria, and like this particular bacteria that you get a slight enrichment for compared to no worm, but not really in the overall like mass of the microbiota, is more efficient at making short short chain fatty acids, or
sure. I guess my point is
on them. But
I want the mechanism of how the helminth is controlling that bacterial population.
Yeah, and if it's going to be consistent, like if you used a different mouse, like a different genetic background, or you used a different worm, or you like had a different diet. I mean, like there's so many things that alter, like anything alters the gut microbiome.
Yeah, it's not exactly the most robust observation, or it's not necessarily the most robust observation. Right.
So I would like to see this extended.
They do go into pigs and humans. Are we just not going to talk about the pigs and humans data?
I don't know.
I mean, they do show that there is an small but increase in short-chain fatty acids in pigs and in humans when they are infected with helminths.
Yeah, that's true.
I'm just throwing it out there.
What if when you're infected with a worm, it actually just controls your appetite and you just really want a ton of oatmeal? You're like, damn, I wish I had some friggin' oatmeal. And like, it's all like this like weird.
Well, but mice don't get a choice appetite in the oatmeal consumption.
Well, what I'm talking about with the pigs and the humans.
Yeah, I don't know that pigs get a huge choice in their dietary consumption either.
I only eat pigs that have the choice of eating oatmeal if they want to.
But are they infected with worms,
What if they just
Matt?
start eating like wood chips that they're housed in? What if there's just like some amount of wood chips and they're like, you know what? I want more wood chips. I'm getting some fiber in my little mouse body.
That's true. I imagine the mouse bedding in cages in animal facilities is pretty high in fiber. That is true.
Yeah, exactly. I thought I would close this paper by telling a story about this one time that I was a worm biologist.
I was too at one time.
It was the 9th grade. I was 13. And I was in honors biology. And the experiment for that week was to do experiments on planaria. To be fair, I feel like these would not actually pass any sort of ethics review. Because there was no clear hypothesis. But the idea was planaria are regenerative. Cut them any way you like and see if you can grow 2 planaria. And
how did it work?
So, okay, well, I think I first should say my test subject's name was Meatloaf. And I had a pretty good idea. I'm not going to brag. I was a pretty smart 13-year-old. I peaked at 13. I was pretty sure the bilateral symmetry cut was going to regenerate the 2 planaria because, to be honest, like, it only has to grow, like, you know, it can just, like, copy itself.
Right.
Anyway,
sure.
If you Wikipedia, that is the right answer, but I decided to be a renegade and to see what would happen, a true scientist, what would happen if I took, you know, the road less traveled and just go pop, hamburger style, cut right in the middle. So then I had meat and loaf. And meat was the side with the eyes, and loaf was the side with no eyes. And I regret to inform you that loaf grew a lot of fungus. Perhaps he had fungi in his microbiota. He was completely gone and fuzzy, which I actually think is pretty poetic for someone named Loaf, or a half of a dead worm named Loaf, to completely be taken over by probably bread molds.
I am cutting all of this because it is so incredibly irrelevant to anything. I mean, I love you, Kate.
I also regret to inform you that meat also died eventually, but he did not fuzz over. He just kind of floated. It was sad. So I actually do have a history with worms.
There's this new TV show where they follow Civil War surgeons, and I imagine they had the same scenario play out with all of their amputated arms.
I feel like this should be kept in the podcast. This is very important information for our listeners to know that I do have a slight training in the field of helminths.
Is right, and if anyone has ever tried radial symmetry, please write in. We need to know.
I should probably just get in touch with some people from my old biology class. I bet someone did just cut it into like 4 pieces.
I think we've lost Kevin.
I would
like to edit this episode.
You're welcome to do that. Fine. Okay, so
well,
just to sum up,
This has been a meandering episode
Somewhat.
of Audiommunity.
But not too long. It's been a meandering but concise episode.
That's true. That's true. This is a fairly straightforward paper, I feel, because they sort of repeat a couple things. But basically, take-home message, worm infections alter microbiota, microbiota alters immune function. I kind of wonder if all of the effects of the hygiene hypothesis that we see with worms, like people have had these hypotheses that worms induce type 2 immunity, and you have this shift in the balance towards Th2 versus Th1 or Th17, and that's what's causing reductions. I wonder if all of it's just the effects on the microbiota. That seems a little implausible, but I guess you can't really rule it out. I don't know.
No, I think I would actually be surprised in the other direction. I would be surprised if it wasn't all... I mean, I certainly think that there's going to be an immune-centric component to all of this, but I don't know that you can consider that even without considering how that interaction between the immune system and the microbiota is setting itself up. And I think necessarily if you change one side, you change the other.
I agree with that. And I also think that there's probably a very strong genetic component.
Right. Well, that's almost certainly true. There is with all these things. MHC haplotype is always the top hit in any GWAS study around autoimmune disorders and allergy.
Yeah, okay.
It's actually the only one that ever is significant.
Okay. You know what, though? Look, I have a bone to pick with GWAS studies.
Okay.
Slightly because I'm not the biggest fan of large data sets, but I mean, I like GWAS studies and the idea that they have the ability
or
to
animal models generally.
You know, there are only so many ways that we can really study human genetics, and it's a very complicated field.
So let's just do everything in vitro.
So, I mean, no, we can't have scientists that are like cynical like me. We need like scientists who are who want to do the animal studies and deal with uncertainties and like kind of statistically significant on the edge of significance studies. We need those people. It's God's work, you know? But okay, GWAS studies. The biggest hit is always MHC.
Yes.
It's the
It's not even close.
Okay. What is the word I'm looking for?
Diverse?
Yes, it is the most diverse allele. I mean, there's so many alleles. The
but
idea
it's usually
that
it's usually one specific allele that shows up, though.
I know, but it's the only gene that there are so many alleles for. So, like, of course, one's popping up. There's no other diversity.
But it's the same one that comes up over and over again.
Those
like
B
the
types?
the same alleles show up over and over again, unfortunately, in these populations.
B7, 57, 37.
Well, there's different ones for
11.
for infection versus like autoimmune disorders versus, but yeah, I mean, yes, it's the most genetically diverse, but you're not just looking at genetic diversity, you're looking at like SNP diversity and stuff. So you can have small alterations that don't rise to the level of the crazy diversity of MHC that would still come out. And I don't think the way that the statistics work, I don't think that just having diversity is going to have an effect if that diversity doesn't actually correlate. Like if you don't have a selection towards one particular allele, it's not going to show up.
If you have such a rare, if you have a super, super rare genotype, you're not going to have a lot of people who have it to like, I mean, I'm sure this is all like I'm sure this is all taken care of in the statistics, and I just would have to go to like that GWAS course and learn. But just intuitively, I don't like it.
That would be tragic. I think I have to know more to actually have an intelligent opinion on this, but I'm going to go ahead and say my opinion anyway.
I'm just going to say it. I understand that I should learn about this to have a more informed opinion, but I think that like the GWAS studies, if there's not like an inherent amount of diversity, you're only going to hit the diverse genes and the most prevalent, like the highest amount of diversity, because everyone is diverse in those genes. There's a lot of like motion going on, right?
Yeah, there's more variation in to sample.
Yeah.
Is what you're saying. There's more variation to sample.
Exactly.
Therefore, it is more likely that you're going to find something.
Exactly. Whereas if you just have like 99% of the human population, you know, has like the same allele and like 3 people don't, you might not hit them as like being important. That's all. I'm sure this is something that GWAS people know and it drives them mad at night. Mm-hmm. I'm sure that I've just hit like the most important question.
Yeah, you're probably right. There is no GWAS biologist that is getting any sleep ever because this troubles them in their dreams.
it does. I mean, like, just thinking about the statistics of like what you've run, and you're like, have I really done like the most robust analysis of like all the different variables? And like, you're just like, oh my goodness, there's like so much sensitivity analysis to do, and you just stay late at night. Actually,
I can tell you that actually Chester Alper seemed like this bothered him quite a bit.
I'm actually being dead serious.
Yeah, no.
Well, with the nightmares of GWAS biologists, this has been Audiommunity.
Are they biologists?
Recorded on February 1st, 2016. I'm Kevin Bonham with Matt Woodruff and Kate Franz, and I don't want to find the paper that has the whole spiel, so I'm just going to wing it as I often do and say that you can find us at emmunity.org, emmunity.org, and on Facebook and on our website, and comments at emmunity.org. I did go submit us to Stitcher, so you can subscribe to us on Stitcher now, as well as iTunes, and also Google Play doesn't do podcasts, but you can subscribe to our RSS feed. You can grab that from our website.
Are you sure? I just listened to a podcast. It was like, you can find us on Google Play.
That's ridiculous. I don't believe that.
I 100% am not lying to you. There's no other way I would know what Google Play is.
Fair enough. I'll look into it. Fine. Facebook, website, podcast, subscription, yada yada. Music at the beginning and end of the show is recorded by Rachel Reineck, and we'll see you next time.
Meatloaf lives. That was a little Mark Maron.
I am cutting that
like,
for sure.
Because he goes like, Boomer lives. Meatloaf was my pet planaria.
I get it.
I got the pet planaria thing. Oh, God. Okay. I'm stopping recording now.
I'm also not drunk, so that's exciting.